Hello friends, I'm Anaka Sabu, first MSc Biology, St. Mary's College, Thrissur. My topic is genes, alleles and variation. Gene is a hereditary factor or hereditary determiner that governs heredity, produces variation, controls metabolism, directs development and regulates growth and reproduction. It consists of contiguous segment of DNA. In eukaryotes, a gene occupies a specific position in a chromosome called a gene locus. Genes are four major classes. One, structural genes which encode for particular protein and engineer its production through the production of mRNA intermediate. So the structural gene can undergo both transcription and translation. Two, rRNA genes which do not code for protein and cannot undergo translation but can undergo transcription to produce rRNA. tRNA genes which do not code for protein and can undergo only transcription to produce tRNA. For regulatory genes which are not transcribed and translated but serves as recognition signals for enzymes such as DNA polymerases, RNA polymerases, etc. Alleles or allelomorphs are alternative forms of a gene which occupy identical loci or homologous chromosome and control the contrasting forms of same trait. Usually a gene existing in two allelic forms, dominant and recessive. Dominant allele would be expressed always, but recessive allele is expressed only in the absence of dominant allele. This is because the dominant allele would mask or suppress the expression of recessive allele. Dominant allele are usually symbolized by capital letters and recessive alleles by small letters. Variation. Adaptation and diversity are two central themes of evolution. It's common phenomenon that no two individuals are alike. Even the identical twins are found to differ from one another. The difference between closely related animals are called variations. Variations has universal occurrence. All individuals exhibit differences as well as similarities. Each human individual differs in quantitative or qualitative aspects of voice, shape, body, gestures, and behavioral postures. Variation is the raw material for evolution. All variation can be grouped into 6 types. They are meristic variation, 2 substantiative variation, 3 continuous or fluctuating variation, 4 discontinuation variation, 5 somatic variation, and 6 germinal variation. Meristic variation refers to the difference in the number of body parts, structures, etc. Example, presence of 6 fingers instead of usual 5, presence of 13 ribs instead of usual 12 in man. Substantiative variation, this is the qualitative variation where differences occur in the shape, size, color, etc. one of the animal and their part. Example, color of the hair, eye, skin, shape of the hair, nose, etc., height, etc. Continuous variation are small or minute differences. Individuals in a population are continuously different from one another with intermediate forms. They exhibit statistically a parametric or normal distribution around the population or sample mean value. Darwin stressed on the small variations. He called them fluctuations. According to Darwin, new species originate by the gradual accumulation of small variation. So there is possibility for the occurrence of intermediate stages. Discontinuous variation. These are large and appear suddenly without an intermediate stage. Darwin termed these large changes as saltations and he believed that they are useless for evolution. De Vries believed that these sudden changes are important for evolution and he called them mutations. Individuals in a population are discontinuously different from one another with no intermediate expression of a particular character falls either into one category or another. Example, hair lip in man, presence of 6 finger, ancient breed of sheep. Polymorphism is a good example for discontinuous variation. Human ABO blood group is suitable example to illustrate discontinuous variation. Each human is either AB or O. There are intermediates. Alleles at a single chromosomal locus determines the ABO antigen for specific individual. Example, hair lip in man, presence of 6 finger, ancient breed of sheep. 5 somatic variation. Characters which appear on the somatic cells are called somatic variation. They are developed by the influence of environmental factor. Lamar called them acquired characters. Somatic variations are not heritable. Germinal variations. Character appear in the germ cells are called germinal variation. As the germ cells are transferred to the offspring, germinal variations are heritable. Genetic variation is important for evolution. According to Mayer, non-genetic variation adapts the individual, while genetic variation adapts the population. Source of variation. Variation is produced by a number of factors. They are the follows: gene mutation, chromosomal aberrations, recombination, hybridization, isolation, natural selection, genetic drift, founder's principle, and migration and gene flow. Thank you.